Welcome everyone. Today we're diving into Module 3, Enzymology. This is Study Session 3, Enzymology 1, as part of BCHM 241, which covers the biochemistry of macromolecules. I'm excited to guide you through this topic, where we'll explore the fundamental principles of enzymes, how they work, and their importance in biological systems. So let's get started and uncover the fascinating world of enzymes together. Let's dive into the fascinating world of enzymes as catalysts in biological systems. Enzymes are remarkable proteins that speed up chemical reactions, making life processes possible at the speeds necessary for survival. Now, an important aspect to consider is cofactors. These are non-protein molecules or ions that assist enzymes in their activity. Without cofactors, many enzymes would remain inactive or less efficient. Moving on, we'll look at how enzymes are named and classified, which helps us understand their specific functions. Generally, enzyme properties include their specificity, their ability to work under mild conditions, and their regulation mechanisms. When we talk about classes of enzymes, we're referring to the main categories based on the types of reactions they catalyze, such as oxidortictases, transferases, hydrolases, and others. All these points come together to highlight the complexity and elegance of enzymatic functions, central to biological systems and crucial for maintaining life as we know it. Enzymes are chemical substances that play a crucial role in catalyzing biochemical reactions. Essentially, they speed up these reactions by lowering the energy barrier needed for bond formation, which makes the process more efficient. The first enzyme ever isolated was urease, and this breakthrough was achieved in 1926 by James Sumner. It's interesting to note that, apart from ribozymes, which are RNA molecules capable of catalytic activity, all enzymes are proteins. An important feature of enzymes is that they contain an active site. This active site includes specific residues. These are amino acids that participate directly in the reaction, and often a cofactor, which is a non-protein chemical compound that assists in enzyme activity. Understanding these components helps us appreciate how enzymes are finely tuned to facilitate life's essential biochemical processes. Let's take a closer look at the active site. This is the specific region of an enzyme where the chemical reaction actually takes place. Think of it as the enzyme's business center, where all the action happens. The active site has a unique shape that perfectly fits its substrate, kind of like a key fitting into a lock. This specificity is crucial because it ensures the enzyme only catalyzes the right reaction. When the substrate binds to the active site, it forms an enzyme-substrate complex, which then lowers the activation energy needed for the reaction to proceed. This is what makes enzymes so efficient and vital for biological processes. Understanding the active site helps us see how enzymes control and speed up reactions in our bodies, tying into the bigger picture of how biological systems are finely tuned for life. Let's talk about some key properties of enzymes that make them so fascinating. First, they have a high catalytic power, meaning they can significantly speed up chemical reactions that would otherwise take a long time. This efficiency is one of their most important features. Next, specificity is crucial. Enzymes are highly selective for their substrates, which helps ensure reactions occur precisely and efficiently. It's also important to note that enzymes do not change the overall reaction equilibrium. They just help the reaction reach that equilibrium faster without shifting the balance itself. Their activity, however, isn't constant. It can be influenced by factors like temperature and pH, for instance. If the temperature gets too high, enzymes can denature, losing their shape and function. Similarly, pH levels can affect enzyme structure and activity. Lastly, substrate concentration plays a role too. As substrate levels increase, enzyme activity generally increases until it reaches a maximum point, known as saturation. All these properties, high catalytic power, specificity, stability within certain conditions, are essential for enzymes to do their job effectively in biological systems. They help maintain the balance and efficiency necessary for life processes. Let's take a closer look at how temperature and pH influence biological systems and chemical reactions. These two factors are crucial because they can significantly affect the structure and function of molecules. For example, many enzymes have an optimal temperature range where they work most efficiently, and if it gets too hot or too cold, their activity drops off. Similarly, pH levels can alter the charge and shape of molecules, impacting how they interact or catalyze reactions. Understanding these parameters helps us grasp how living organisms adapt to their environments and how we can manipulate conditions in industrial or laboratory settings to achieve desired outcomes. So, when studying biological processes or designing chemical experiments, paying attention to temperature and pH isn't just a minor detail. It's essential for ensuring accuracy and effectiveness. This connects directly to our overall theme of understanding the delicate balance required for optimal biological and chemical function. Let's look at some examples that illustrate these concepts in action. These examples help us understand how the ideas we've discussed can be applied in real-world situations. By examining them closely, we can see how the key points come together effectively. Remember, the goal is to connect theory with practice, so these examples serve as practical illustrations of the principles we've covered. As we go through each one, think about how they relate to the overall theme of the presentation and how they might inform your own work or decisions. Let's talk about the substrate. The substrate is essentially the foundation or the base material that supports the entire system we're discussing. It's a crucial component because it influences the performance, durability, and overall functionality of the final product. When we think about substrates, we need to consider their material properties, such as conductivity, stability, and compatibility with other components. For example, in electronics, the substrate might be a silicon wafer that hosts the circuitry. In biological applications, it could be a tissue scaffold that supports cell growth. The key point here is that choosing the right substrate is vital because it impacts how well the system performs and how reliable it is over time. Connecting this to our overall theme, understanding the substrate helps us optimize the entire process, ensuring that the system functions efficiently and effectively. So, whenever we're designing or analyzing a system, paying close attention to the substrate can make a significant difference in the success of our project. Initially, enzymes were named based on the names of the individuals who discovered them or by adding the suffix "-ase to their substrate. For example, lipase for lipids, nuclease for nucleic acids, and peptidase for peptides. However, these naming conventions have limitations because they don't provide enough information about the specific type of reaction or the substrate involved. To address this, in 1961, the International Union of Biochemistry, or IUB, developed a more systematic classification system for enzymes. This system assigns a 4-digit commission number to each enzyme, which helps standardize their identification. Enzymes are classified into 6 major classes, and each class is further subdivided into 3 subcategories. The major classes are designed to represent the type of reaction the enzyme catalyzes, giving us a clearer understanding of their function. This classification system is a crucial part of enzyme nomenclature, helping us communicate more effectively about enzymes and their roles in biological processes. Now let's talk about classification. Essentially, classification is about sorting data into categories or groups based on specific features or characteristics. It's a fundamental concept because it helps us make sense of complex information by organizing it into understandable segments. When we classify data, we're applying certain rules or algorithms that determine which category a particular data point belongs to. Think of it like sorting emails into spam or not spam. It's about recognizing patterns and applying those patterns to new data. This process is crucial because it underpins many applications, from spam filtering to medical diagnosis and even credit scoring. It connects directly to our overall theme of making data-driven decisions, as classification enables us to predict outcomes and automate processes with greater accuracy. So, in summary, classification isn't just about labels. It's about empowering systems to understand and categorize information in a meaningful way, which ultimately helps us make smarter, faster decisions. Now, when it comes to myocardial infarction, or a heart attack, one of the key indicators we look for is an increase in the levels of certain enzymes, specifically creatine kinase, or CK, and lactate dehydrogenase, LDH. These enzymes are normally present in our tissues, but when damage occurs, such as in a heart attack, they are released into the bloodstream, making their levels rise. The interesting part is that these enzymes exist in different forms, or isoenzymes, which can tell us more precisely where the damage is happening. This is incredibly useful because it helps us diagnose whether the damage is specific to the heart or perhaps another organ. So the clinical significance here is that by measuring these enzyme levels and identifying their specific forms, we can gain valuable insights into tissue or organ damage, which guides our diagnosis and treatment decisions. This approach ties into the overall theme of understanding biochemical markers to assess health and disease more accurately. Now, let's wrap up with the clinical significance of lactate dehydrogenase isoenzymes. As we saw earlier, these different isoenzymes of LDH are quite useful because they help us identify which specific organ might be damaged. Each isoenzyme has a unique tissue distribution, so when we see elevated levels of a particular isoenzyme, it points us toward a specific organ or tissue site of injury. For example, LDH1 is predominantly found in the heart, so its elevation can indicate myocardial damage. Similarly, LDH5 is more common in the liver and skeletal muscle, so increased levels could suggest liver injury or muscle damage. Understanding these patterns is crucial in clinical practice because it allows us to narrow down the possible causes of a patient's symptoms and guide further diagnostic testing. Connecting this to the overall theme of enzyme diagnostics, LDH isoenzymes serve as valuable tools for pinpointing the location of tissue damage, making them an important part of our diagnostic arsenal. So, recognizing which isoenzyme is elevated helps us determine the affected organ and tailor our treatment approach accordingly.